You think taste happens on the tongue. It doesn't. The tongue only detects signals. Taste is constructed, interpreted, inside the brain. What you call flavor is one of the most complex sensory illusions biology has ever created. For decades, textbooks taught a taste map of the tongue: sweet at the tip, bitter at the back, salty on the sides. That map is wrong. Modern research shows that all basic tastes can be detected across most regions of the tongue. There are no isolated zones, only differences in sensitivity. The tongue is not a keyboard. It is a distributed sensor surface. Taste perception begins in taste buds, microscopic sensory organs embedded in papillae. There are three main papillae involved: fungiform, scattered, especially anterior tongue; foliate, lateral tongue; circumvallate, posterior tongue. Each taste bud contains 50 to 100 specialized cells, constantly regenerating every 10 to 14 days. Taste is short-lived. The system is designed for renewal. Taste cells are not neurons, but specialized epithelial sensory cells. They convert chemical signals into electrical ones. Different cell types respond to different taste modalities: sweet, salty, sour, bitter, umami. Each modality follows a distinct molecular logic. Sweet, bitter, and umami use G-protein-coupled receptors, GPCRs. Binding activates gustusin, phospholipase C, calcium release, and neurotransmitter secretion. Salty and sour are different. They rely on direct ion channel flux. Salt alters sodium flow. Acid alters proton concentration. Different chemistry. Same goal: electrical signaling. Once activated, taste cells release neurotransmitters onto sensory nerves. 3 cranial nerves carry taste: facial nerve, V; anterior tongue; glossopharyngeal nerve, X; posterior tongue; vagus nerve, X; epiglottis and pharynx. Taste is already being integrated before it reaches the brain. Signals converge in the nucleus of the solitary tract in the brainstem. From there, information is relayed to the thalamus, the brain's sensory gateway. Taste is filtered, weighted, contextualized. True taste perception emerges in the gustatory cortex, primarily in the insula and frontal operculum. But it does not work alone. Taste merges with smell, texture, temperature, memory, and emotion. Flavor is not a sense. It is a neural synthesis. When smell is blocked, up to 80% of flavor disappears. Taste buds still work. The brain just lacks context. Taste without smell is incomplete information. Taste evolved for survival. Sweet for energy, umami for protein, salty for electrolytes. Sour warns of spoilage, bitter warns of toxins. Taste is not pleasure first. It is risk assessment. Genetic variation alters receptor expression. Some people detect bitterness intensely. Others barely sense it. Taste is personal biology, not preference. Taste does not exist on the tongue. It exists in interpretation. Every bite you take is a decision made by neurons, based on chemistry, memory, and survival. You don't taste food. Your brain does.